Professor Andreas Börner, he is very active in plant breeding, as you may know, and he has a very dense and compact scientific achievement in this area. So if you allow me, I just briefly mention some of the important key elements of his CV, just as a glimpse of the knowledge what he did beforehand. Because not me is the main person today, however, is Professor Andreas Börner. So I just want to introduce him as an excellent scientist in plant breeding, and we are really pleased that he was elected by the Academy as a honorary member. So Andreas Börner received his PhD in plant breeding in 1988 in Martin Luther University in Halle, Wittenberg, and then he, between 1990 and 1996, he used to work in the Leibniz Institute and working on plant wheat genetics and the breeding program. Then he was as an expert in the IPK or IPK. He really was the responsible person for maintaining the gene bank, and he was responsible for develop especially the serial gene bank and resources, including not the cultivated, but also the wild relatives or wild relatives of this. So his scientific work was mainly based and did excellent job on the genetic and the resources and the gene bank maintenance, and also in this respect. Later he was as a professor, he became a professor at the Martin Luther University, and he was very active in the teaching program of the university. He supervised more than 18 PhD students and 71 MSc or BSc students. So the next generation of the plant breeders and the genetics may be close to his scientific achievement and guidance. In the Web of Science, we listed out that he has more than 330 scientific papers, with more than 8,000 citation, and also his Hirsch index is 46. So he became editor of the Serial Research Communication recently, and we are pleased that this is one of the key, let's say, connection with the Hungarian breeders. As the Serial Research Communication, as you know, this was initiated and started in the 80s, I guess. And now he's the editor-in-chief of this very important journal in this field. Also, he's serving several other journals as editor or working like Applied Genetics and Hereditas, and we can list more than the important scientific journals in this field. Currently, his major work is that he is the president of Eucarpia, which is, you know, that this is the most important organization of the plant breeders in Europe. He also is working in the ISTA, which is the Seed Testing Association, which is also having a very important role in plant breeding and in the genetic resources of this area. He was actually honored by the Life Sciences University Lublin in Poland as a honoris causa doctoral degree, and as I mentioned, he was elected as member of the Hungarian Academy last year. So please, Andreas, give your inauguration lecture. Thank you.
Thank you very much for this very kind introduction. And before I start with my presentation, I would like to say that this is a really great pleasure for me to be here today and a great honor to become a member of the Hungarian Academy of Sciences, because I have a very special relationship to Hungary. In fact, my international scientific career started here nearly 35 years ago. So a visit to Hungary, to Martonvasar and Szeged was my first, as I said, my first connection with international people in research. And as you know, the first love is always the deepest. So I'm very happy to be here. And I come back to this a little bit later. Now I would like to start my presentation, which is entitled Plant Genetic Resources for Food and Agriculture, the Backbone of Feeding Future Generations. This sounds really a big, it's a big title, but it is really true. And I would like to show you this, that plant genetic resources are very important for future plant breeding, mainly. And I will divide my presentation into these 5 parts. I will give you first an overview about plant genetic resources on a global scale, what's going on the conservation of this material. Of course, I will talk about the German ex situ collection at the IPK in Gartersleben, where I'm from, and I will follow with giving you examples on research, and at the end I will mention several examples on the successful German-Hungarian collaboration. Let's start with the global collections. You see this map here. So it is estimated that there are 7.4 million accessions worldwide stored in our global gene banks. And there are 1,750 of these banks, which are shown here on this map. And the 10 largest ones I did highlight in this light blue color. And the largest ones, these are the gene banks in the US, then a gene bank in Mexico. There is an international wheat and maize breeding institution. The gene bank at the IPK in Gartersleben, it's among the top 10. Then the gene bank in Russia, in St. Petersburg. There was a gene bank in Aleppo, in Syria. You may know there was the ICARDA institution, but due to this war which took place, this collection moved to Morocco and Lebanon. Then the national collections in India, China, South Korea, and the Philippines. So these are the big gene banks in the world. And you see on this map also this red column. And this red column, it's an international seed store at the so-called Svalbard Islands, where the gene banks have the possibility to store their safety duplicates. I will come back to this a bit later. If you look for the numbers, you see here that the largest collection is in the US, followed by China, India, Russia, Japan, Mexico, Morocco, and Germany. And if you look for the crops in these collections, then it becomes clear that the 2 crops feeding the world, wheat and rice, also have the highest numbers of accessions in our global gene banks, followed by barley, maize, bean, sorghum, soybean, oat, groundnut, and cotton. So these are the 10 big crops in our worldwide gene banks. And with that, I move over to the German ex situ collection at the IPK in Gartersleben. And we have here a nice view on the whole institute, which has about 600 employees. And the gene bank department is one of 4 scientific departments which are at the IPK, and it is located here rather in the middle or in the center of the institute. And you see here this more modern looking building. These are our cold store facilities where we are storing the seeds. We store the seeds at minus 18 degrees Celsius. I will tell you this also later. And what you also see on the slide, and you can recognize it, all these glass houses here, large ones, but also a lot of rather small ones, we are using or we need for the reproduction of the material. The small glass houses are for the reproduction of cross-pollinating species, for example. And you also have here a view on our experimental fields, which are also used for gene bank reproduction. If you look for the numbers, then you see that as on the global scale, the cereals have the largest number, followed by legumes, vegetables, oil fiber crops, medicinal spice plants, forage crops, and so on. In total, 150,000. I already did mention this. And these 150,000 accessions belong to 3,000 species and 780 genera. And with that, the German gene bank is also one of the most diverse gene banks in the world besides the US. And beside this collection stored in the cold stores, we have also a very huge reference collection. As you see here, a reference collection comprising 450,000 herbarium sheets, 110,000 samples of seeds and fruits, and 57,000 samples of cereal spikes. I also should mention that in 2008 we did implement a so-called quality management system. That means all processes going on related to storage, related to regeneration, to drying, to threshing, and so on are well documented, so that people always can follow these protocols, even if the stuff is changing. The material goes back to collecting missions. Some of them are listed here. And the oldest material, just to mention here, it comes from the 1920s. So more than 100 years old now, and when people began to collect material. And as you see here, researchers from IPK, they went, for example, to China in the 1950s, to Iran, to Mongolia, to Libya, Iraq, North Korea, Ethiopia. These are regions where nowadays nobody can go. It's completely impossible. But the people at that time did these missions, and this also shows that the material stored in this gene bank have a very, very high value. And this is it's very good for us. So how we store this material? And this is one of these protocols. Of course, you cannot read, you should not read, but here's exactly described how we have to store the seeds. And I will explain this to you. So we have a so-called active sample, which is stored in such glass jars at minus 18 degrees Celsius. If we need seed, we take it out, we open it, take the seed, close it, put it back. Then we have the basic sample. It is stored in aluminum bags under vacuum, also at minus 18 degrees Celsius, because we know today that if you store the seed under vacuum, it will survive even longer. And there is a safety duplicate. In fact, it's a triplicate, also stored in aluminum bags at minus 18 degrees Celsius at the so-called Svalbard Islands. Svalbard Islands, where we find them, and this you see here on this slide. So Hungary is somewhere here, Germany here. This is Norway and Sweden, and even in the north of Norway there are the so-called Svalbard Islands. In German, we say Spitzbergen. And there, a seed store was built in the frozen rocks, as you see here. And inside, there is a possibility to store the seeds. And here is another picture of this seed store, which is exactly here. And as you see here, I had the luck to be there with 2 colleagues. Just in February 2020, it was just 2 weeks before the corona epidemic starts, and it was really cold there, I can tell you. minus 28. We use this store and we send material to this place frequently. As you see here, we have now 67,000 samples on this place stored. Okay, so this was a short overview about our gene bank, and now I come to examples on research. So we are not only storing the seeds, we are also performing research. And one very important question for us, or for any gene bank, but also for all the breeders, is how long does a seed survive? How long we can store the seed? Not forever, but how long? So we initiated work on what we call seed longevity. Seed longevity means how long a seed survives. And we started to work with seeds which were stored under ambient conditions. That means 20 degrees Celsius, 10% seed moisture content. So not the cold storage, the ambient storage. What we found, I will show and explain you now in the case of such diagrams. So what you have here, you see this diagram, and you have here the age of the seeds, and you have here the germination in percent. And the red line is the important one. This is the so-called P50 value. That means germination dropped down to 50%. And in wheat, this is 7 years. Not so bad. In rye, it is 6 years. In maize, it is 12 years. So maize is very robust. Seed of maize is very robust. Keep in mind under room temperature. 2 more examples. Lettuce, lactuca, rather sensitive. P50 value is 4 years, and after 6 years or 7 years, it's gone, it's dead. And another, the last example, pisum. Pisum again, very good survivor. P50 of 14 years, and after 20 years, it still may germinate. So what does it mean? It means there is an intraspecific variation. So there are differences between the species with respect to their storability. Okay, good to know. But when we look here at the pisum slide, we see something else. We see that at the beginning, the variation is rather low. Okay, they are all here in this circle. Later on, there's a huge variation with respect to the germination. So after 15 or 16 years, we still have accessions having 95%. Others drop down to 3 or even 0%. So besides this interspecific variation, there is an intraspecific variation. There's variation within the species, and this makes the gene bank management so difficult. You cannot say we store pisum for such amount of years and wheat for those amount. No, we have this variation within the species, and we have always to check. Okay, so this was at room temperature, but I told you in the beginning, our material is stored at minus 18 degrees Celsius in big cold chambers. These cold chambers were built in the 1970s, and the oldest material is from the harvest 1974. So this is around 50 years now. And we have so-called historical germination data. We always perform germination tests. And we did evaluate and analyze this data. And the results for me were quite promising. So we have here wheat. The diagram is a bit different, but again, the red line is the P50 value. And as you see, after 38, 40 years, most of the accessions are still fine. They are above 50%. So wheat is very good or easy to store. For barley, it looks similar. There may be some decline here. So you have to check at least after 30 years what's going on. But here, everything is really fine. And of course, certain accessions are lower, but this is the normal variation. But if we look now for rye, rye looks different. So it's fine for the first 10 years, but after 30 years, again, we have this huge variation. Some accessions are at 90%, others down to 0. And now you should keep in mind that this material in this particular year, which gives us now this variation, was grown on the same fields in Gartersleben, had the same climatic conditions, the same rainfall, the same soil, of course, with some variation. It was harvested at the same time with the same machines, and it went to the cold storage sitting side by side in the shelf. So the environmental conditions were more or less the same. But why we have such a huge variation here at the end? And our conclusion was that this variation must be due to genetic differences of these accessions. And the idea was to perform genetic analysis. So how to do this? One way is you take this accession here, and you take this accession from here, you make crosses, you develop DH lines or whatever, but then you have to wait for another 30 years till you get this decline in the germination or this variation. All of us who are working in science know we have not 30 years time. We have to publish each year. So, but there was some solution because there are there are methods for so-called artificial aging or accelerated aging. I have no time to go into the details, but if you treat the seed with high relative humidity and high temperature, they age very fast, yes? Or if you treat them with a high pressure of oxygen. So we used such kind of methods and started to perform genetic analysis. We started with barley. We used a mapping population which was already available, which was built for something else. And we did regenerate it in the field. We did the artificial aging here in these glass jars, high humidity, high temperature, and we got a nice phenotypic variation from 0 till 85 or what. So, and from this population we used here, there was already a genetic map available. So we just had to run what we call the QTL analysis. That means, just to explain, whenever you get such a peak here, so this is the chromosome, right? And whenever we get the peak here, that means here must be a gene or a gene cluster or whatever responsible for the trait of interest, and in our case, responsible for the seed longevity. And you see we were able to find such peaks here. And just to explain one of them, this one here on chromosome 7H, this was linked to a gene which is responsible for naked caryopsis. So in barley we also have naked forms. And in fact, the naked forms lived shorter than the hulled ones. So we were able to clarify the genetics of this trait in barley. We did a similar study in wheat, but in wheat we found also loci, the red ones here. And in wheat, we also had, within this population, a difference in seed dormancy. And people were always thinking, or it was not really clear, if a seed has a high dormancy, that means he does not germinate immediately after sowing, does this seed also have a high longevity? So it lives finally also longer. But our results did show that this not seems to be the case, because the gene for seed dormancy were on a completely different chromosome compared to all the others. So we believe these are two different mechanisms. Another study was performed in oilseed rape, as you see here, but also in tobacco. This was a collaboration with some people in the Polish Institute. So this was a nice work we could perform during a couple of years. We got nice publications, as you see here, which came out from this genetic studies. And in the last year, we even prepared some review summarizing all these studies, genetic aspects and molecular causes of seed longevity in plants. So we summarized everything what was available from Arabidopsis and rice, but also wheat and barley. These were our own results: maize, soybean, lettuce, tobacco, with the Polish colleagues, and tomato. Okay, this is nice. But what does it mean? Now we know regions within the genome where we have genes, QTLs, or gene clusters responsible for seed longevity. So there are the genetics behind this trait. But still we do not know what is the reason for the seed aging. It does not really help if we say we have 5 genes in barley which are responsible for the trait without knowing anything about the reason for aging. So we decided to go another way in addition to that what we did. And we started to do what we call metabolite profiling. To investigate the metabolites, we used wheat, also stored at different years, at different temperatures and so on. And we did the germination assay, and in parallel my colleague David Rive, he did the metabolite profiling. And what he got was a very nice picture. Look at this. For me it looks a bit like art, modern art, but what you see here are 20,000 chromatographic features for lipids. So he called this picture the wheat lipidome. So these are all the lipids and oxidized lipids occurring in wheat seeds. And of course we correlated this features, these dots, with the germination. And 11,000 of them were significantly correlated to germination. And one example I give you here. So this shows a really very nice correlation between germination and metabolite content. And the metabolite we have investigated here is the so-called O1-diacylglycerol. Don't ask me what it is, but at least it is correlated to the seed aging and to the seed longevity. And now we try to use such kind of methods in order to determine the germination in the gene bank collection. So the data were published recently, so if you are interested, you can read this, or you can ask me. I can send you the link. So this brings me now to the last but most important part of the presentation, the German-Hungarian collaboration And I told you in the beginning that I was here about 35 years ago, and there was still this letter I found in my hard copy files from March 1989, and there was written, I'm not sure if you can read or not, there was my name is written there, to visit Hungary, and there is written Dr. Otto Weiss and Dr. Gabor Galiba, who I should meet during this visit. And then it took not so long, I guess. So this was in March, and in June I was ready, or I was allowed to go to Hungary. It was still in the communist times, you know, it was in summer 1989. But I was happy to go there. And I still have my notebook from these times. I told you, the first love and the first notebook one should keep. But I don't know why I did keep it, but I still have it in my shelf. And there is written here, Ungarn, so Hungary, 12th of June till 21st of June, and my name. So, and there were also notes about my discussions with Gabor and with Otto, and at that time with Josef Schutka, of course. And yeah, it was interesting to look to this booklet again. So, but what did we do? When doing my PhD, I was working on the dwarfing genes, on the genes for the Green Revolution. Most of you know this. These dwarfing genes were the ones led to shorter straw, more nitrogen could be applied, higher yield could be achieved. And your people here in Hungary, Schutka and Kovac, they described in 1987, so this was even before, a gene which was named RHD12. And they did the chromosomal location. So they found out on which chromosome this gene must be. And here you see this mutant, or this short straw line. And in fact, this short straw mutant were obtained by a gamma ray treatment of a Hungarian cultivar Karsági 522. And due to this gamma ray treatment, the mutant appeared, which was very short, and it was named RHD12, as you see here in the paper. And the mutant was then named Karsági 522M7K. And the Hungarian colleagues, Schutka and Kovac, they made a monosomic analysis. So what is a monosomic analysis? This was, at that time, very high sophisticated research. There were monosomic series, as you see here, of two cultivars, China Spring and Cheyenne. They were crossed with the mutant, and based on the segregation of the progeny, you could identify the chromosome. And it was chromosome 5A. Good. At that time, one could not do more, but I was very much interested in this mutant. And after I have been here, I also received it, and a few years later, the molecular markers appeared. And in that time, mainly the so-called wheat microsatellite markers. And as you see, we were able, what we say, to map, to identify the region of the gene on the chromosome. And Schutka and Kovac were right, it was chromosome 5A. So we found this gene on the distal part of this chromosome. And at that time, I also did work on rye. And in rye, there was a similar gene on chromosome 5R. And the idea was, or the question was, is it similar? Is it the same gene or not? And doing further mapping studies, we could really show that RHD12 and the rye DDW1 seems to be so-called homologous genes. So the same genes in the same regions on the genome, and this was later on also published. And the interesting thing here was that, as I told you, RHD12 was induced by gamma rays, so it was a mutation, man-made mutation, but the Rye gene was a natural mutation. It was found in the gene bank in St. Petersburg. And this was really interesting. By these two different mutations, we seem to have identified the same or highly comparable gene loci in wheat and in rye. So let's continue. And this was the time when Andreas Börner visited me in IPK. I'm very happy that he's here today. And he was working on heavy metal tolerance. I never worked on it before, but I was thinking, okay, let him do this. And we analyzed so-called substitution lines. And what does it mean, substitution line? Just for those people who do not really know, so we have one cultivar, in this case Chinese Spring, which was copper tolerant, as a recipient. So the close to violet chromosomes here, and we have a donor cultivar, Capelle de Pré, copper sensitive, and we have 21 lines where we always have one of these chromosomes substituted. And based on these lines, and these lines were tested, and based on these results, we can identify chromosomes, or we could identify the chromosomes having these genes. And just to show you here, we have the two parental lines. Chinese Spring has 100%, and Capelle de Pré has only 88% tolerance. And checking all the lines which were available, you see we found certain lines which has significantly lower tolerance compared to the Chinese Spring, to the recurrent parent. And when we looked a bit more in detail, it turned out that 3 of them were localized on the homologous group 5 chromosomes, so 5A, 5B, 5D. So the conclusion was the group 5 chromosomes should carry genes for copper tolerance. In addition, we were able to have wheat-rye substitution lines, and we could identify also chromosome 5R carrying genes for the trait of interest. So again, the homologous group 5 chromosomes of wheat and rye contain loci for copper tolerance. So this was the main result of this research. But we did not stop, we did continue. And there was another paper. And you see, I always have underlined the Hungarian colleagues in red and the German ones in blue. So, and again, this was Andras, it was Gabor and Fruzsina at that time. And what we did here, we developed so-called single chromosome substitution lines. This is a special genetic stock where you have recombination only for one chromosome. The rest is identical. And this enables you to find genes on this special chromosome. And again, it was chromosome 5A, as you see here. And these single chromosome recombinant lines were developed by Josef Schutka at that time. And we analyzed them with the molecular markers which were available in the lab of Marion Röder. She was the leading person in developing so-called microsatellite markers. And we were able really to find QTLs on this chromosome 5A. And the interesting thing was that one of them was in the same region as the vernalization response gene. And this was somehow surprising. We checked also deletion lines we had available, and again we found a gene for copper tolerance very close linked to the vernalization response gene. And the question appeared then is, does VRNA1, so this is the vernalization response gene, have a pleiotropic effect on copper tolerance? This was the question at that time, and it's still the question today. It's not solved yet. But anyway, this was interesting. Now, the last trait where we did collaborate on together was drought tolerance. And this was a big experiment. It was led by Fruzina Gira, and also Gabor and Andras were included, and several of my colleagues again. We did tests in the germination stage, in the seedling stage, and in the adult stage. And the material was grown in Hungary in the phytotrone part of the plants. It was grown in the rain-out shelter there, and there were a huge work was done. I remember this very well. And what was analyzed was a mapping population. And I will not go into detail. This is one of the tables we found. And just to summarize this, there were 5 main clusters identified on chromosomes 2H, 5H, 6H, and 7H, where QTLs were stable over at least 2 developmental stages. Yes, and this was really a very interesting result. Another joint publication appeared in 2008. And there, this is very interesting, there Fruzina did compare different methods, screening methods, at different developmental stages in barley. So seedling stage, germination test, hydroponics, so seedling test, and then field tests, and so on. And what she describes here, it's still valid today, and not only for barley, but also for wheat, I think also for all other crops, for people dealing with such kind of tests. So the germination test, of course, is quick, it's simple, it's cheap. These are the advantages. Disadvantage, of course, not reliable for predicting adult stage tolerance. So, and all these things, different things were described here. The last example I give to you today of our collaboration was published 2 years or 3 years ago. It was again drought response, drought tolerance after chemical desiccation or the use of a rain-out shelter. And the rain-out shelter was placed here in Hungary, in Martonvasar. And the Hungarian colleagues did this work here. We did the chemical desiccation. What does it mean? I will tell you in a minute. So what we checked was a set of 111 spring wheats, and with a very modern, high-dense marker set, the so-called 15K SNP array. That means 15,000 markers were used for this population. Remember, when I started with the monosomic analysis, we were able just to identify the chromosome. Then we had the microsatellites. We could find the position on the chromosome, but several Sandy Morgans always away, and now we have 15,000, of which 13,000 did work finally on the whole genome, so we can do a very accurate mapping. The material we investigated, this 1,100 spring wheats, came from different areas of Europe and Asia, and also here from South America. And what we did was chemical desiccation. What does it mean? So in Germany, usually we have no severe drought during summer. But if we want to test the plants in the field, we have to use some method. And we use the chemical desiccation. What does it mean? You have a plot, you divide it into two parts. One half is sprayed with a calcium iodide solution, the other half is untouched. And after 2 weeks, you see how it looks like. This is the control, this is the treatment. So the chlorophyll is gone, and the plant can only make yield by mobilizing stem reserves. And you see also the grains here. Look, the grains after the desiccation are shriveled. Of course, you may say that this is not a really drought test, that's right. But in the late 80s, correlation studies were done for this method, and climate chamber experiments, and the correlation was by 0.8 or so. So we did this. The main reason was why we did this, that I had this student here, Khalil. He came from the Iran, and he had he was paid by the Iranian government, and he was paid for studying drought tolerance. So we had to do something with drought, and we did it. Anyway, so this is the result. I will not go into detail, just look at the two red circled arrows here. These were the two loci which were found in the rainout shelter experiment in Marton, Russia. I'm at the end. I come to the summary, and the summary is just one slide showing you all the joint publications we did, we could produce during this time, during the recent years. Again, you see the red and the blue lines, so it's a good mixture. And the work is not finished yet. We just are now again in discussion with Gabor to do some work, again related with the Green Revolution genes, the RHT genes. And with that, I would like to thank you for your attention. And I have here two photographs which are interesting to me. So this photo here was taken in 1989 in Marton, Russia. You see myself here in the back, a rather shy person, not in the front, in the back. You see Gabor here. He was not so shy at that time. And here you see a photograph from the 2022 with myself. And you see that the phenotype did change slightly, yeah? But the genotype is still the same. Yeah? And with that, I thank you very much for your attention, and it was my great pleasure to be here today. Thank you very much.
So my last duty today to hand over the certificate of the Academy, and this is written in 2 languages, both in Hungarian and in Latin. I won't read out the Latin text because I don't know how many people are speaking Latin here. I know that some of us learned Latin in the secondary school, but I dare to read out because I think it's just the important is that this is the diploma that Andreas Börner became elected as honorary member of the Hungarian Academy.
Thank you very much.